For about the past decade, European and U.S. government agencies and advocacy organizations made a declaration that our goal was to achieve to find disease-modifying therapies, meaningful treatments for Alzheimer's disease by the year 2025. That was a goal that we set as a field a little over 10 years ago. But to achieve that goal, we realized that many things needed to change in the ways we do clinical trials and the efficiency in the ways we do that. So in this talk that I gave, I emphasized some of those things that had to occur and how far along that has brought us towards our goals to 2025 approvals of disease-modifying therapies. About half of the talk was focused on biomarkers, because in fact, biomarkers have played a key role in improving the way we do clinical trials and identifying the right patients for the right drugs at the right stage of disease for a clinical trial. About 10, 15 years ago, when we started doing anti-amyloid therapy treatments, for example, in phase 2 and phase 3 clinical trials, we quickly learned that if you only use clinical diagnosis for enrollment of patients into clinical trials, you would end up with a good proportion of patients that didn't have the targeted pathology, whether it be amyloid, whether it be tau, whether it be Alzheimer's disease in general. So very early in the process, it became fairly standard to start using amyloid PET scans to identify patients to ensure that they had amyloid in their brain. And throughout the talk, I demonstrated different ways that biomarker development has evolved and is now contributing to designing more efficient clinical trials. Some of them, like I said, we started with using amyloid PET, but not too much later. Tau PET came into the picture, and now there are trials that are utilizing tau PET to identify participants that have clear tau pathology, because tau is more tightly linked to clinical progression and underlying neurodegeneration. So if you identify patients in the right stage that have enough but not too much tau, that might be the ideal population for proof of concept phase 2 studies to test your efficacy hypotheses with molecules. But now we're getting into a stage of development where we're starting to have fluid biomarkers as well. Spinal fluid has been utilized clinically for many years now, but there are access issues. There are with spinal fluid. So development of plasma biomarkers has really been a tremendous goal of the field to improve access and efficiency in trials. So now we're starting to see clinical trials that are using combinations of both tau PET or amyloid PET with plasma biomarkers, plasma biomarkers of either amyloid or tau to enroll into clinical trials. So maybe using a plasma biomarker to pre-screen to identify people that would most likely meet your criteria for PET if, for example, PET was the key entrance criteria into a trial. But now we're starting to see trials that are utilizing plasma biomarkers by themselves to try and identify that right population, that homogeneous population to enter into clinical trials, to give those clinical trials the best shot on goal at showing an efficacy signal. And I think that's very important, particularly in phase 2 studies.